Okay, good afternoon, everyone. And this afternoon, we are just going to finish off our discussion about DNA and its use in forensic science. It's a very important section for you to understand because this is really one of the areas of forensic science which is going to only be developed more and more and more and more as time goes by. Just remind you where we finished off. We're talking about the current ways of using DNA, which is to use what are referred to as short tandem repeats in the DNA sequence. Short tandem repeats are small segments of DNA. Just give me 1 second. I just want to try and do something. There we go. The short tandem repeats are very short segments of repeating bases. In this case, we look here, AATG, AATG, and those segments repeat over and over and over again. Now, what we're looking at here is a particular locus, a particular place in the human genome. These are two matching or paired chromosomes, one from mother, one from father. And as you can observe, there's an area here where these short tandem repeats occur. And this has a specific name. This is called FO1. And at the start here, before these repeats begin, there will be a known sequence of DNA that we can make a primer for. That means we can direct polymerase chain reaction at this area, and we can amplify this FO1 area specifically. Out of the whole human genome, we can head straight for the FO1 sequence segment and start to amplify it. Point 1. Point 2, there are a number of variants of this segment in the human population. I believe there's about 8 or 9 different variants, and they vary by the number of segments that are repeated. So here we see that there are 6 in this segment here. In the paired chromosome, however, inherited from the other parent, let's say this is mother, this is father. Mother had donated a chromosome with 6 of these segments. Father donated a chromosome with 8. So this is shorter than that. Now, when we do the PCR, there's just a little added thing to tell you about PCR. And that is that we don't just sequence this strand here. We'll actually sequence both strands at the same time. We make a primer to sit there, and it will replicate that. And we make a primer for here in the matching strand, and it will replicate that section there. This is very important because it's I don't want to go into all the reasons why, but you just need a round or 2 of the polymerase chain reaction. And what you end up with is you only end up amplifying the area between those 2 primers. Okay, so for the mother's chromosome, FO1, we'll end up with a fragment that is this long, right? When we look at the father's, it's 8, but it's got the same markers, it's got the same primers. It'll have the same primer here at the start, and we'll duplicate that. It'll have the same primer here, and we'll duplicate that strand, and we'll end up duplicating between the 2 primers. We'll end up duplicating a fragment that is that long, longer than that. Because this is longer than this one here, we can actually take our DNA sample and we can somehow run it in a system which will discriminate those fragments. It will allow them to pull apart from one another and must allow us to visualize them. And when we do so, but there's various ways to do it, but the classic way is to run it out on an electrophoresis gel. And here's a gel electrophoresis of exactly this kind of pattern that one would see. And we've loaded our DNA samples here. This is the gel, and we've put an electrical field across, positive down here, negative up here. And because DNA is negatively charged, it moves from the wells, it moves down towards the positive pole. But as it goes, the gel retards the DNA molecules, and it slows them down. But the small fragments, in other words, our 6 fragments, for example, will move faster than the fragment which is 8 repeats long. And so they'll get separated out from one another, smaller down here, larger here. Okay, so we go to the crime scene, and there is a DNA, there's a source of DNA there at the crime scene. We take that DNA and we will subject it to polymerase chain reaction, and we will amplify the Tho1 site. Remember that in any DNA sample from a human being, we're going to be sampling on 2 chromosomes, one from the one parent, one from the other, and the number of repeat segments can differ on the 2. So when we look here, we're seeing the site from one parent and the site from the other parent on the down below. Now, this here is the DNA from the crime scene, and you see it has 2 bands. We have 3 suspects, so we get DNA samples from them. We do exactly the same treatment, PCR, using the same primers, etc., and we will amplify the Tho1 site. Look at this one here. Something weird has happened because this suspect here has only 1 band. What does that mean? It means that the maternal and paternal number was the same. In this case here, we would designate this as Tho6-8. In this case here, we would designate it as 8-8. Okay, so oops, sorry. So this is 6, this is 8. If we look here, 6-8, that single band was larger than the bottom band, right? So that must have been 8. This is 8. That there would be 8-8 on both maternal and paternal. The next suspect, look at this suspect here, and you see he has identical banding there. How about this here? Totally different. The one site is much smaller. It's smaller than 6, and the other is larger than 8. So clearly we exclude that suspect. We exclude that suspect, but we do not exclude that suspect. This does not prove that this is the perpetrator. It simply confirms our suspicion, because this is only one site. That's only one STR site. And if we look at the percentages, we can work out what the frequency of these different sizes are in the human population. And because we know that, we can work out the probability that a matching like this, crime scene suspect, we can work out what are the probability that a matching like this occurred by chance. Well, in actual fact, for a single site, it's quite high. If we look here, that particular pattern, Tho168, is actually found in 3.5% of the population. That's not a small percentage, right? That means that this analysis can successfully exclude these suspects, which is very valuable. And it provides corroborative evidence that this suspect should continue to be examined, should continue to be looked at. But so that for one site like that, we've got good class evidence, not individual evidence. But the great power of the single tandem repeat analysis like this is that these sites, different sites with different structures with different repeating sequences occur throughout our genome. They occur on all of the chromosomes practically. And many of them have been identified together with their little flanking areas where we can make PCR primers so that we can amplify those particular sequences. So because of this, once we have one site and we know its probability of chance occurrence. Then when we look at another site, we work out its chance of its probabilities. We actually multiply those probabilities. And we begin to move from really broad class evidence to very, very definitive individual evidence, because it ends up, when we use lots of different sites and combine their probabilities, the probability of such a combination occurring by chance is often infinitesimally small, 1 in billions or trillions. So the more of these sites, these single tandem repeat sites that we can look at, the more powerful the evidence becomes. And so in the it used to be that they used 13, so these are the figures for 13 sites. Now 20 sites are used, 20 different sites. But if we just take the 3 commonest sites, THO1 would be one of them. But if we took the 3 commonest SDR sites, worked them out, and then worked out the probability for all 3, we would find that it's about 1 in 5,000 people could have that same combination of the 3, and you could pick it up by chance, 1 in 5,000. If we match 6, we get to 1 in 2 million. If we match 9, we get to 1 in a billion. If we match 13, there's a little bit, it gets very definitive. We begin to look actually at ethnic probabilities at this level. You begin to look at ethnic probabilities as well, which does influence it. If matching 13, 1 in 575 trillion Caucasian American, 1 in 900 trillion African American. More people than have ever lived. More people than have ever lived on the planet. So when we match 13 sites, we are able to stand in court. You do not, the DNA expert does not say, this must be the person because I say so. The DNA expert says the chance that DNA recovered at the scene and DNA recovered, the standard DNA examined from a suspect, the chance that those were purely by chance are 1 in 575 trillion, 1 in 900 trillion, even higher with 20 different sites. It is extremely powerful evidence, which we can regard as being individual evidence. So remember that we've only had about 100 billion people have ever lived. We currently have 7 billion people, and we're talking about 1 in 575 trillion chance of a match being by pure chance. When they run these kinds of analyses, when we looked here, this has been run singly. Okay, this is just the DNA from a single site. It'd be very tedious to repeat these gels over and over and over again like this. So instead, sites are specifically chosen so that the primers for them are radically different to one another, completely different. And as a result, many sites can be run by PCR at the same time, and they can then be discriminated, and the fragments, the sites are chosen, so the fragments they produce are very different for the different sites. So each of these, this here is one particular single tandem repeat site. This is another one here. This is another one here. And you can run several at the same time. And this is called multiplexing. And this is added to this the system is speeded up by running it through a capillary tube system rather than a gel system. It works in much the same way. The capillary that they are run through, the little fine tube, is specially treated so that it retards the DNA molecules and does the same thing as the gel, retards the larger ones more than it retards the smaller ones. And we can discriminate them in the same way. So this now, much of this is done by robot in laboratories. Once the samples have been prepared, they can be run by robot. This also has another advantage. And doing it by technology, such as a robot system, means that you duplicate the technique exactly from one case to another. There's no bias. Unfortunately, even in DNA, in this DNA typing, malfeasance has sometimes entered into it, where technicians have, or whoever, have been determined to show that somebody is guilty and have swayed results one way or another by essentially forging. When you use a system which is completely blind to any of those kind of considerations, you can have higher confidence, in fact, in your results. The robot also speeds things up enormously. And this has been one of the major disadvantages of the advent of DNA technology, is that many crimes can produce DNA for possible typing, but it all has to be done. It has to be done by, it has to be accomplished. And tremendous backlogs in specimens have developed in many laboratories because laboratories are simply unable to keep up with the demand. That has been hugely improved by putting them through these automated systems. So as I've told you, we use 20 STRs. And not only do we have the advantage now that we can do so many single tandem repeat sites with great efficiency, but also the technology has improved so much that we use less and less and less is required, and really only trace amounts of DNA are needed, and hopefully in those that we need those sites that we test to be intact. But in fact, the DNA itself can be degraded, provided we have enough single tandem repeat sites intact that we can analyze them. So this tells you the 125 picograms of the DNA, 125 billionths of a gram of DNA is required with current PCR. PCR does have limits to it. There are limits to the amount that you can start off with, but there are also limits to the number of times that you can run, that you can repeat. You must realize that if you're doing a polymerase chain reaction, should an error occur early on in your replication, that error may be replicated over and over and over and amplified along with the DNA. So there is a very great need for good technique, but also for quality control in the DNA laboratory to ensure that the technique is as good as possible. This amount of DNA is about the same as 18 cells. Very, very little. Very, very little indeed. Is essentially a trace, and could certainly, if somebody held something like a weapon or something like that, far more skin cells than that would be rubbed off on the surface. If somebody bit something, either chewing gum or somebody's skin, the saliva they left behind would contain far more than 18 cells. So that would be easily amplified from there. Okay, so this is just There is a section on the automation and everything if you want to read it in your text, but this is the kind of output you'd get from a capillary system, and it tells you the size of fragments at each of particular. Here's SO1, by the way. There, in this case, was 6 and 9. I don't know why 9.3, but in any case, it's there. That's SO1. This one here, 27 and 30, 15 and 16, 10 and 13, etc., etc. And when we look at these, just realize that everybody in the human population could have different numbers at these different sites. And when they have progeny, they will donate one number to their child. The other parent will donate one number, and so we very quickly build up a system where this becomes highly individuated to a particular individual. Okay, so just one or two other kind of aspects to DNA analysis. There are methods using PCR where we can do sex determination, even if we are unable to otherwise determine the sex. The biological sex is determined by whether or not you have a Y chromosome present. And that is one pair of our chromosomes, which often do not match. These are called the sex chromosomes, and there are two kinds. There's an X chromosome and a Y chromosome. Females have two X chromosomes, and males have one X chromosome and one Y chromosome, and they inherit the Y chromosome from their mother. Daughters inherit one X chromosome from mother, one X chromosome from the father. And because the Y chromosome is inherited from the father, and the father inherited it from his father, etc., 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 the Y chromosome becomes common in a system of paternity. Everybody derived from a single paternal figure who has a Y chromosome has the same Y chromosome, on and on and on back through your grandfather, great-grandfather, your grand-uncles, your great-grand-uncles, etc., etc. You all have Y chromosomes. If you're a male, you have Y chromosomes, which are the same. If we go to if we find a Y chromosome and we type it in any way, because there are areas of the Y chromosome that can be typed, it gives us class evidence. It ties the Y chromosome to all of those other Y chromosomes that are identical to it in that paternal line. So it gives us some information, but it is relatively limited information. There are single tandem repeats located on the Y chromosome, but there are a couple of special things about it. And first of all, there have been more than 400 have been identified. That is actually not a tremendous number, considering the size of the human population. The one time that it becomes very useful is when multiple males are involved or could have been involved in a sexual assault. And this is one way in which they can be sorted out, is by looking at their Y chromosomes, looking at the SCRs on their Y chromosomes to see if they differ. So Y chromosomes are inherited through the male line, but I think I've already told you that our mitochondrial DNA is inherited actually through the mother, through the maternal line, all of us. When fertilization occurs, the sperm contributes only its nucleus to the fertilization process. It does not contribute any of the cell contents. There's one little tiny organelle which it contributes, but that just does not concern us. It does not contribute mitochondria. The sperm has mitochondria, but they do not enter the fertilized egg. Instead, we all inherit our mitochondria, which are the powerhouses of the cell, from our mothers. But the mitochondria are extremely interesting little bodies, and they actually behave in some ways almost like independent living things. And one of the reasons for this is that they have their own DNA. They have circular chromosomes of DNA. They have a single mitochondrion, may have several, but they're all the same. And it also, by the way, has its own ribosomes to make proteins. It doesn't make all its own proteins, but it makes some of its own proteins. So its DNA is active. Its DNA is used to produce proteins in the mitochondria. But the mitochondrial typing, again, because we inherit it through the maternal line, we all share our mitochondria with our siblings and then with our maternal relatives. So we share mitochondria with our mothers and our mothers' siblings and our grandmothers, etc., etc. So we end up actually sharing our mitochondria with often quite large numbers of people. Just think, for example, I'll tell you that if you go back 10 generations, you have more than 1,000 great-great-great-great-great-great-grandparents. And if you go sort through them, you'll be able to find out which one of them contributed the mitochondria, which comes all the way down the generations to your generation, and you share with everybody who inherited those mitochondria. So mitochondria give us good class evidence. It performs the same functions class evidence always does. It may inform our conviction that somebody should be looked at further. It does not prove guilt, but it often may serve a useful purpose in excluding people as well who do not share the same mitochondrial characteristics. The other huge advantage of mitochondrial DNA is that it can be sequenced. It can be typed for very old DNA. And it is, in fact, even been typed from Neanderthal skeletons and fossils and things like that. So it survives very well, partly because it's a small molecule, and also probably because it is enclosed in the mitochondria in another membrane system, and it gets preserved quite well. There are two areas on mitochondria which are called hypervariable areas, and the hypervariable areas are very, very short little sequences of DNA, which do seem to vary a lot in the human population. But I think about 4,000 or 5,000 variants have been identified, which is far more than the number of variants of mitochondrial DNA. But they're not very useful. It's extremely tedious to use them because they have to actually be sequenced. All the systems that I've told you about, the STR analysis, RipFlip, these sort of systems, they're not looking at actual DNA sequence. They're looking at DNA structure, not the actual sequence. To sequence DNA, to get the order of bases, A, T, A, G, G, A, C, is a rather tedious process. And to do it for mitochondria is even more tedious. However, there is still ongoing research attached to the use of mitochondrial DNA in forensic science, and methods for using mitochondria DNA is improving. So that's something you may hear about in the future. At the moment, it provides class evidence. I think I've told you all about this. Let me just check that I have. Yes. Okay, so the other major advance that has arisen out of DNA, using DNA, is the establishment of this fabulously valuable DNA database called CODIS, which is the Combined DNA Index System. Now, CODIS is a repository for DNA typings, which have been determined from many different sources. It depends state by state, it varies as to whether or not you are obliged to give DNA, for example, if you are arrested. The Supreme Court kind of regards DNA donation in much the same way as they regard the requirement for providing fingerprints, that it is simply a metric, that it is not illegal search and seizure, unreasonable search and seizure to demand a DNA sample. But states do vary in their legislation as to whether it's allowed. But in most cases, people convicted of a crime are required to provide DNA. And all of those go into the CODIS database, along with DNA which has been volunteered. And people volunteer their DNA under many, many different circumstances. Even, for example, even if you need to read fine print carefully on many things like commercial DNA kits and things like this, because that may be made available to the FBI. At any rate, it's currently is close to 1 million seq profiles, DNA profiles, are deposited from unsolved cases in CODIS. And what they are waiting for is for the same type, same DNA typing to be picked up, maybe at another crime scene or under another circumstance, submitted to the CODIS database and get a match. Likewise, there are many cases where a DNA typing is done, it is submitted to CODIS, and somebody is positively identified from the CODIS database. So it can work both ways. In addition, as you probably know, DNA evidence is very, very long-lived, and it was often inadvertently collected. For example, in a rape case in the 1960s or something like that, there was no such thing as DNA typing. It had never been thought of. But evidence was collected, clothing, sheets, whatever, with prominent DNA staining, semen staining or whatever, blood, whatever, and has been kept for all of this time. And now you can go back and re-examine. The DNA can still be typed, even from very old samples, provided it has been carefully kept. And many such cases have been solved by typing old evidence and then looking in the CODIS database and being able to put the two together. A very famous one at the moment is the Golden State Killer case, where years and decades ago, this man stopped killing in California. Everybody assumed he was dead or something, but in fact, he was still alive, and he was found by examining old evidence, and he was identified through DNA typing as an very, very elderly man. If you want to look that up, it's the Golden State Killer. I think you probably even find it on Wikipedia, something like that. Okay, so DNA evidence is tremendously important. It has to be extremely carefully collected. The various sources are fairly obvious: blood, semen, saliva, touch DNA, as I described, from holding something. Touch DNA implies that even a fingerprint will give you enough cells. That is unlikely. It's usually, touch DNA is usually from a firm hold on something. It could be from a surface, but it's more likely acquired from a weapon or something like that, that the person has actually held or held firmly, like a doorknob. that would be quite possible. Hair, it's a mixture of things. Hair by itself, hair that has simply been shed, will seldom yield a DNA profile, but it may yield a mitochondrial DNA profile. Hair which has been pulled out, which has a skin tag, a follicular tag on it, that is extremely valuable. That is very rich in DNA. And then DNA can also be recovered from bones, especially from teeth. And the quality of the DNA will vary a lot, but it is certainly possible, and it has been done, that DNA has been recovered from skeletons, skeletal remains. I'd also just point out there's other uses for DNA as well, and pure identification is often important. A body which is unrecognizable, skeletal remains, all of these, they can be identified because we can type their DNA and then we can compare it to family members. And if you compare the DNA to family members, they will share many of their STR sites, and the typing can prove or disprove a relationship between them. The other use, of course, is in paternity testing, where paternity is disputed. DNA typing can definitively prove or disprove paternity. So now investigators always examine a crime scene with an eye to possibly collecting DNA evidence, because it's such a powerful technique. And anything, bottles, cans, cigarettes, a bite mark, anything like that, which might have been touched or used by a perpetrator or by a suspect, would be examined, and swabs taken, and then the swabs amplified for DNA typing. Or clothing is very important, because so much rubs off on clothes. So many skin cells, so many body secretions accumulate in clothing, as it's often a very, very rich source of DNA. Any blood evidence, and blood evidence can be collected even when it's dried. The DNA is usually absolutely fine, particularly if it's relatively recent. And DNA, any of these samples, unless it's really, really profuse, they're usually dried in order to be kept. And if they have to be kept for a long period of time before they end up in the laboratory, they are kept under refrigerated conditions. Standard samples, that is when you have a suspect and you require DNA sampling from them, it is usually relatively easily done, because all of our cells in our bodies contain exactly the same DNA. They all contain exactly the same DNA. So if you are wanting to compare a blood stain collected at the scene to the DNA from a suspect, you don't have to take their blood. You can take any of their cells, and that usually means taking a swab from the inside of the mouth, which is a very rich source of cells and a very rich source of DNA. The collection of DNA has to be done extremely carefully. First of all, it must be solidly tied to the site where it was collected from. Because the techniques are so sensitive, it could be very easy to slip in a DNA sample that has been collected by malfeasance. And this has been done. That investigators are convinced somebody did something, and they trick them into donating an inadvertent sample, which they slip in as evidence. It's unfortunate. Human beings think of all sorts of devious things, but if the evidence is carefully collected at the scene, if it's photographed and documented, then its origin can be proven in a court of law, and its chain of custody, it becomes incontrovertible. The packaging of anything with with DNA, usually dry it as quickly as possible. And the drying does not hurt the DNA at all, especially if the drying is done relatively quickly at a reasonable temperature. The other thing is that it's very important not to contaminate samples. One of the huge difficulties in dealing with DNA is if a DNA sample is actually mixed. If the mixing is clear, in other words, for example, if you had a case of rape or something like that, where you had DNA from a victim and DNA from a suspect mixed in your sample, it is possible to sort them out because you have two reference samples. It becomes extremely difficult where there's no reference sample.
Professor?
And yes, go ahead. Yes, what did you want to ask me? I can't hear you. I think you're muted.
Oh, when is the final paper due?
Please just save your question for the end, please.
Okay.
Not while I'm in the middle of discussing something.
Okay.
I'll discuss it with you at the end of class, all right?
All right.
When you're collecting DNA evidence like this, it's really important not to contaminate, because if you contaminate with an investigator, you're introducing DNA which might never be traced. Nobody will ever think, oh well, instead it sets investigators off on a wild goose chase to find somebody unknown. And sorting out DNA samples which are mixed with unknowns in them is extremely, extremely difficult. Now, I'll just refer you, in your textbook, there's a very short paragraph on page 413. And it's entitled Probabilistic Genotyping Software. So this is a raging controversy in forensic science at the moment. It's always been known that dealing with mixed DNA samples is extremely difficult. And it's one area where it becomes subjective. Especially if some of the DNA is unknown, because the investigator is looking at, you can imagine, it's two superimposed patterns, and the investigator has to decide what belongs to who, and it becomes subjective. So very often that evidence was very difficult to use in court. And a laboratory came up with a very, very elaborate algorithm to examine mixed samples and provide what they said was an unbiased analysis, statistical analysis, which would separate the samples, and everybody was thrilled. It was used in several cases before actually statisticians got hold of it and ripped it to shreds and said that that it just could not work the way they were describing that it worked. And many of those cases now have got to be re-examined. Anyway, read the paragraph, but be aware when you read that paragraph that it has come under considerable controversy and is not used. Should not be used as it was developed. Okay, so anybody collecting DNA, they're wearing gloves. They usually now dressed in personal protective equipment, not to protect themselves, but to protect the crime scene. Lab coats, gloves, eye protection, etc., etc. And what they're doing is limiting any source of DNA that could contaminate evidence. And obviously it is cleanly collected, put into clean receptacles, and that is usually paper or cardboard, which breathes and which will allow the specimen to dry out rather than sit and fester and grow mold or whatever. And that's about it. Okay, so sorry, I interrupted you now, but you can ask me your question. You want to know when your final paper is due? Your final paper is due the end of this month.
Okay, and what do we have to write about?
It's there in Canvas. The instructions are there on Canvas for you.
Okay, and do we have a final for this class as well, or no?
You have a final for this class, and the final consists of 2 parts. One is a class test. One half of the final is a class test, and the other half is a comprehensive test of material that you've already seen.
Okay.
All right.
Okay.
Okay then. All right then.